my
Are you
slide
able to
here.
advance the slides?
Well, I've still got Chris' slides up on my screen.
Yeah,
So I advance
that's
the
what we
next one
see.
now. Okay. Then
Laura, can you help advance?
I can, yeah, I see the little thing now. Okay.
Okay.
So that,
Yeah,
okay. We're
great.
getting dual advance. Let's go back one maybe. And I'll briefly stop being antisocial. I'm actually out here. But you probably don't need to look at me. We can focus on the slides. So if we go back one, maybe we can start. If I can control it, then I will do that. Yes, looks like I can. So what I want to do here is kind of try and re-emphasize a lot of the messages that have come out of the other talks. So you'll see a bit of duplication, and that's fine, because in essence, some of the messages are key. I'll try and bring them out. And I'll probably build a lot of what Chris just said to try and emphasize that, alum's been around forever. It remains the present, but it also has a significant future in vaccine development. And I'll try to illustrate how that's going to happen. So it looks like there's a lot of build in this slide. Hopefully, it's okay. So yeah, same history as everybody else. Of course, Alexander Glennie was the first to use, maybe since he's a Brit and I'm a Brit, I can call it aluminium, but then I can abbreviate it to alum, even though we've already established that we're not really using alum. It's just a helpful abbreviation. But alum adjuvants have been around an awful long time, originally used for toxoids and subsequently included in many different vaccines. Yeah, my advance doesn't seem to be working. Oh, there we go. Maybe it's just slow. And this is kind of emphasized on what you've heard previously. There's the different adjuvants we now have, but for pretty much the whole of the 20th century, we only had aluminium salts, and they were used in many vaccines. They continue to be used in all the childhood vaccines around. Safe and effective. Gradually, others have emerged. MF59 was mentioned. That emerged at really the turn of the 21st century. And ASO4 also emerged soon after. And that's something else I'll talk about, because in essence, it's a next generation adjuvant built on the basis of alum salts. So newer approaches become available increasingly in the last decade or two, but aluminium salts remain utilized in many vaccines. Sorry, there seems to be a lot of build in this slide, which is not really necessary. So some basic points about alum salts and other adjuvants. Safety is established, been used in billions of doses in many different populations over many decades. Increased reactogenicity at the injection site is common for any adjuvanted vaccines, including alum-based ones, relative to injecting a protein alone. But the vaccination-related symptoms, generally mild to moderate, short duration, and do not impact compliance with vaccination schedules. So generally, you get more reactogenicity with adjuvanted vaccines, including alum, but they have an acceptable benefit to risk ratio in all the populations in which they're utilized. And this is kind of, some of these things have been mentioned during the talks. This is going back a decade or so, where with some colleagues, I tried to establish a rational approach of how you do R&D around adjuvants. And, I think this pretty much still applies. And the starting point is, of course, well, don't use them unless you need them. And there's lots of examples of next generation antigen design where perhaps the antigens are becoming more inherently immunogenic, but often you still need adjuvants. But the need is complex. It depends on many factors, and it may be obvious in preclinical models, much less obvious when you go into humans, and which population, the age of those individuals, whether they've had prior exposure to the pathogen, whether they are immune competent, etc., including elderly. So there are many factors in humans that impact whether the adjuvant is really helpful or not, and you need to determine these. What we always say is, well, Aluminium salts are established, safe and effective and inexpensive. So if you think you need an adjuvant, start there, because it's much simpler if you can accomplish what you need to accomplish using the basic adjuvants. And this was mentioned earlier. I think Chris Harmon and I got together and tried to explain how to use adjuvants, alum adjuvants, most effectively. And if alum is not good enough, maybe oil and water emulsions are a better option. They're often more potent. And then MF59 was the first, and pretty much there's generic versions of MF59 available from various sources. So oil and water emulsions are the second most established adjuvant. So if the alum can't do it, maybe emulsions can do it. Maybe some issues of instability to worry about. But you need to be aware of them in relation to alum too. And then, yes, there are more novel approaches available, particularly in the last decade or so, as I've highlighted. But they need to be fully justified in that pretty much you use them if the established ones don't work so well. And it's going to be more complex. You're going to need to explain the mechanism. You're going to need more data in engaged agencies, often longer timelines as the agencies do the necessary regulatory scrutiny of those novel adjuvants. So don't use complex ones if the simple ones can do the job. And I'll mention as we get on, and Chris already did, the idea of alum plus building something off the basis of alum is a pretty good idea, and it's starting to become more and more established. And this is a brief summary. I think it's actually in the paper. I think we can thank Harm for this one. And in essence, it's an important message when you think about the utilization of aluminium adjuvants. It's complex, and you've heard various people talk about it. The manufacturing is particularly complex. Maybe leave that to the experts. But the utilization is also complex. And you need experienced formulation scientists to work through the approaches to enable you to succeed with aluminium-based adjuvants. And we've already heard a lot of academic teams add A to B. That's not optimizing alum, conclude that it doesn't do the job and move on to the much more complex things. And I think there needs to be more work on making sure we utilize aluminium adjuvants in its optimal fashion. And again, I'm singing my own praises to some extent, but I think Harm and Chris wrote more of this than I did. But it gives you an overview of the optimal use of aluminium in vaccines. So it's a good how-to guide. And this is one of my own papers, which I'll use to highlight a couple of important messages, which have already been highlighted previously. So adsorption to aluminium, we've changed to aluminum here, but that's okay, often enhances the stability of antigens. And that can be really important because ultimately we're all trying to create products, and we need products in the optimal form. That can be pre-filled syringes or it can be vials, but you want vaccines that are able to be distributed around the world and at least be fridge stable. And this was an example of utilizing some recombinant antigens that are actually present in Bexero product to show that we could stabilize those recombinant antigens through adsorption to aluminium hydroxide in this situation. And there are many examples other in the literature. And of course, there are opposing examples where you can destabilize the antigen, which comes back to the point we made previously. Work with knowledgeable formulation scientists if you hope to succeed in this space. And this, again, has been mentioned previously. Bob covered it to some extent. Generally speaking, the characterization of vaccines, particularly vaccines including adjuvants, can be complex. There are many alternative adjuvants mentioned earlier that have their own problems, but of course, alum-adsorbed vaccines have their unique, distinctive problems, and often to do with, there are many proteins, low concentration, complex matrices. But in essence, you need perhaps to get the antigen back off the alum, and that's not always trivial. And the concept of aging and changing of the adsorption behavior over time is well known. So the analytical challenges can be complex. You need standardized antibodies, ideally monoclonals, and some of these factors were included. Some of this was debated in a recent review that I did again with harm, and that's highlighted at the bottom if people want to read about the more about the characterization challenges. And this, okay, there's some build in this again. Let's go to the end so everything's there. So this is a publication from I get a decade and a half ago, and in essence, it was a way to perhaps improve the characterization approach. So this was using flow and antibodies that are labeled to directly quantify antigen absorbed onto alum. So the paper, the reference is highlighted below. You can do it with multiple antigens. You can multiplex. Obviously, it's all dependent on the quality of antibodies. But this was, we perceived, an advantage whereby you don't need to dissociate the antigens. You can start to quantify directly absorption of antigens on the alum itself. So we thought this was a step forward, which may be advantageous in a variety of situations and can be multiplexed. There was a follow-up paper that showed how that could work. And this adjuvant has already been mentioned, ASO4 adjuvant system. And this really is the kind of the grandfather of the concept of alum plus, the idea of, well, alum does a good job, and sometimes it's not as potent as you'd like it to be. Often, with particularly soluble recombinant glycoproteins, you need something more potent. We talked about maybe you want to go the emulsion pathway, and this is the alum plus pathway. So this was talked about previously. MPL is monophospholipid A, detoxified derivative of LPS. The molecule is to the right. You can see the phosphate, or you can use the phosphate to adsorb onto aluminium hydroxide, and this is utilized in Ceverix. And in essence, Chris showed this data earlier, but this is establishing the principle that I mentioned. If you intend to use a more complex adjuvant than the established, alum will show that the more complex is justified in terms of level of performance. And you show at the top here, simple comparison of ASO4 in yellow versus aluminium hydroxide. And you show enhanced antibody responses, colors change below. The aluminium hydroxide is in blue, ASO4 is in yellow, but in essence, illustrating clearly enhanced antibodies and memory B cell responses. And there's lots of data, particularly on covering breadth, which adjuvants, the TLR agonists in particular, are good at, and showing that you actually get to cover a lot of HPV viral strains not included in the vaccine as a consequence of the inclusion of the MPL. So breadth is an attribute that's important in lots of pathogens, and next generation adjuvants can help you there. And this is a kind of a brief summary of a paper that, again, Chris highlighted this one earlier. Once it was established the TLR agonists like MPL could be effective adjuvants and could be utilized to make vaccines, we decided that, okay, well, maybe we can come up with a better approach utilizing different TLR agonists. And we knew TLR7 was susceptible to being triggered with small molecules. So we did a screen, and this back in Novartis days, now intellectual property of GSK, that explains how we did rational design, high throughput screens, looking at small molecules. We call them small molecule immune potentiators, SMIPs abbreviated, to discover novel adjuvants that were TLR7 agonists. And yet the paper title was Better Adjuvants Through Chemistry. And this is all published if people want to dive into it and get more detail. And this kind of explains why we did what we did. In essence, so you see at the bottom, the adjuvants available at the time were typically natural products or synthetic versions. And, with TLR2 and TLR4, the MPL, what you see, large, complex, heterogeneous structures with complex structure activity relationships. TLR4 has to come from growing, so it's a biosynthetic process with chemical treatment. You see a saponin, QS21, which is also a natural product, or you see a TLR9 agonist, which is a highly expensive oligonucleotide that needs to be synthesized. And then you look at the bottom right, a TLR7 agonist, a SMIP, a small molecule immune potentiator. So, you can very quickly see this idea of small, simple synthetic molecules is attractive, minimize supply production challenges. They have, discrete chemical identity, no heterogeneity. You can make pure agonists, and the structure can easily be manipulated. In essence, that's what we did. We added a phosphonate to make it absorb to alum. The analytics are real simple. Everyone knows how to do HPLC on small molecules. And we thought, as a large pharmaceutical company, we'd had many years of development of drugs, and we can build on that experience to say, let's utilize all that experience to develop new adjuvants utilizing these small molecules. And I think you've already guessed where I'm going next. There was a extensive debate internally. Okay, we've discovered these molecules and we're able to manipulate them chemically. So we can we can put them in emulsions, we can put them in liposomes, we can put them in nanoparticles. How should we deliver them? And then we decide for clinical progression, well, why don't we just use alum? And the reasons are summarized here, and pretty much it's the summary of this meeting, because alum-based adjuvants are well established, inexpensive, safe and well tolerated. They're known by the regulatory agency, so there's a degree of comfort with them. We already know that they can allow delivery of antigen, and if we absorb the SMIP onto the same alum, then we've got co-delivery of the both. And we can then use these new drugs with existing vaccines if we just add the SMIP to an existing alum-absorbed vaccine. And in essence, the proof of concept was already there, because ASO4 was alum to deliver a TLR4 agonist. So, okay, let's see if we can do the same with TLR7. And again, already highlighted, the use of alum potentially enables stable single dose or pre-filled syringes, which is, desired pharmaceutical forms for successful product development. And, just giving a bit of history here, this is saying when the approach of alum plus the next generation of adjuvants was first described, and these are the products that have emerged. So GSK was first described, the ASO4 was first described by GSK and over 20 years ago, and ASO4 has been included in HPV and HBV. In fact, alum CPG was first described by Coley Pharmaceuticals, which was subsequently bought by Pfizer, but CPG on alum was in fact licensed relatively recently. Then it was Dynavax in partnership with Valneva. The AS37 adjuvant that I just described, the absorption of a small molecule to alum, we described that in 2014. There's now 2 phase 1 trials that have been undertaken, and Chris has already talked about his approach in AAHI with a joint TLR7/8 agonist, 3MO52, also absorbed onto an aluminium hydroxide after you've created a nanoparticle with phospholipids. And I believe it's been in phase 1 trials for HIV and COVID, but Chris can answer those questions. And yeah, at the bottom here, Chris mentioned this. A somewhat similar approach was included in a licensed vaccine in India during COVID. So this approach accelerated very, very rapidly as a consequence of the COVID pandemic. And this is, I think, probably is one of my favorite studies. So I show it in most of my presentations because it illustrates a number of key points. And first of all, I always need to thank Bob Cedar and NIH. This was a collaboration with Bob and his team. And essentially what they were doing were asking, okay, can we utilize soluble glycoprotein from HIV to evaluate which may be the best adjuvants for potent neutralizing antibodies? And this is non-human primates. It's groups of 6, it's small groups, but it illustrates important things. First of all, soluble glycoprotein, poorly immunogenic. Use alum and you make it more potent, but it's still not particularly potent. You can use a TLR4 agonist and you make it more potent still, just in terms of the induction of neutralizing antibodies. Or you can use a TLR7 agonist, which is the AS37, and that again increases potency. So this is head-to-head evaluations in non-human primates. Pick winners, but winners are complex. Oh, sorry, I need to get rid of that one. MF59 is in there too, which was pretty potent for neutralizing antibodies. The TLR7 or TLR4 didn't make MF59 any better. ISCOMS was here. This is Iscomatrix, which is now licensed also in a COVID vaccine. And poly-ICLC was perceived as the gold standard by Bob particularly for Th1 responses. So you can look at antibody levels, and then you can do lots and lots of different readouts, which Bob and his various connections enabled us to do. They were particularly interested in antibody-dependent cellular cytotoxicity, so they wanted to look at the functionality of some of these antibodies. And they could also look at system serology and the glycosylation of the antibodies. And so you see here, even in this relatively small group of animals, 6 non-human primates, the addition of the TLR7 agonist gives a significantly enhanced response over alum alone. But even you see the protein alone is poor, and alum is an effective adjuvant. And it's, which is necessary to enable a successful vaccine. Well, this is a complex problem, HIV. Maybe none of them can, but alum works, and some TLR agonists on top work better. And it depends what you want to accomplish, of course. This is functional antibodies. And then this is looking at the T cell profile. And sorry, this is published. You can see the publication down below. Joe Francica was the lead on this. And this is interesting. So color scheme you see to the right, the black is the gold standard for Th1 responses. This is looking at ELISpot, gamma interferon responses, and the gold standard does what is expected. It gives potent Th1 T cell responses. The ISCOMS is pretty good too. Actually matches the gold standard, and so does the alum TLR7. That's here. So again, TLR7 gives you Th1 responses, and not surprisingly, the protein is down here, the alum is down here. So alum is not a potent Th1 response in these non-human primates, and MF59 is here. It's a mixed Th1, Th2 kind of adjuvant, but you can use TLR agonists to get a much more potent Th1 response, should that be perceived as necessary for the particular vaccine you're trying to make. And I think I've got a couple more examples of studies, and again, the same idea, trying to pick winners, but it depends on what you're trying to accomplish, and it depends on the antigen. So this is another collaborative study. Bill and Melinda Gates Foundation for funding this one. The antigen was completely different here. It was a nanoparticle engineered by Neil King at University of Washington. And this was looking at alternative adjuvants and with different outcomes. Here, this is looking at neutralizing antibodies. Simple oil and water emulsion wasn't great. In fact, alum was pretty good. I think this is 6 non-human primates again. But in this situation, adding TLR agonists didn't really help. So alum alone was a good adjuvant for this particulate. Oil and water emulsion with squalene, so with squalene and tocopherol looked the best, but that's fine. That's ASO3. But here, alum looked good and looked better than the emulsion, and the TLR agonist didn't help much. And then when you looked at protection, it was pretty much the same. Alum alone was good, and then the TLR agonist didn't help much, and the ASO3 was good, and the simple squalene oil and water emulsion wasn't. And again, this is published, so people can dive into this data and take a closer look. But the potency of different adjuvants and what's the most appropriate depends a lot on what you're trying to accomplish and the immunogen that you're utilizing. And so finish the AS37 story. This is looking at data, looking at the transcriptome response, and it's comparing alum TLR4, and alum TLR7. Because simply put, if TLR7 does the same as TLR4, then we don't need it because we've got plenty of TLR4 agonists. This is looking at non-human primates, that study with Bob Cedar. This is 24 hours transcriptome response. You see modules upregulated that show inflammation. So it's a very different innate response to the TLR7, which you see over here, which is more an interferon type response. And then do preclinical models predict clinical outcomes? Well, this is a completely different vaccine, but it's Menc on alum versus Menc with alum TLR7. And this is looking at exactly the same modules in humans and seeing that in essence, you get the identical innate activation, the interferon signature, as you did in the non-human primates. And the question now is, well, what will that innate signal do in terms of the adaptive response? And that's what we're looking at currently. And so I will try and conclude. Excuse me, I'm losing my voice here. And this relates to what I've had to say and also what some of the other guys have had to say. First of all, aluminum, whatever you want to call it, or alum for short, they are safe, effective, and inexpensive. And they often enable liquid, stable vaccines with an acceptable shelf life. They enable product development. That's crucial. However, sometimes they lack sufficient potency, particularly for soluble, recombinant, highly purified antigens. Sometimes you need a more potent adjuvant than alum to enable a successful product development, but make sure you utilize alum first and determine you don't need it before you move on. Make sure you've got formulation scientists helping you use it optimally. And then if you want something more potent, utilizing alum as a solid base for alum plus is a good way forward. You've seen AS04, you've seen AS37, you've seen alum CPG, you've seen alum with 7-8 absorbed in different formats. It seems to be a growing approach for next generation vaccines, bringing all the positive attributes you get from the utilization of the alum base. So my suggestion is that these alum-based adjuvants, which have already been included in licensed products, and likely more will follow. And I think I've just about used up the appropriate amount of time. So thank you very much. I can deal with questions, but I believe the rest of my colleagues can also deal with questions however they come.
Thank you, Derek. I think all the talks have been awesome because it's really taken me back to memories from all the work that I've been doing over the years at Wyeth Pfizer with multiple different vaccines, aluminum, developing of the aluminum adjuvant, plus working with so many different adjuvants and from preclinical studies, taking it all the way to clinical. So this has really been a great, presentations and putting it all together in perspective. So we'll open the floor to questions and discussion. I know we have lost a few people because a lot of people in the East, it's very late for them. So especially China, Korea, places like that, it's kind of late. So it's open. Please be free to ask questions. I'll start with with my own experience with several adjuvants. What we find that in preclinical studies in mice, um, or even in non-human primates, sometimes don't translate to humans. Is there anything that you want to comment on that from a perspective of different adjuvants? Why or obtaining the right models to evaluate various adjuvants.
Yeah, I mean, I think that it's been said repeatedly, preclinical models are not good predictors of human performance. Nevertheless, they're necessary. We need to use them, and we will continue to use them while being fully aware that, it's put simply, everything looks great in mice. As you move up, whatever you want to call it, a food chain is probably not appropriate. Things tend to look less good, but no model replicates the situation in humans because humans are complex. First of all, we're very much outbred, no question about that. But then there's many, many factors. The genetics of humans and the immune response is complex, the diversity of humans, but also the accumulated history of exposure to many pathogens. And the pre-existing immune response is a significant factor in what to expect when you try to use a vaccine. And then even beyond the genetic diversity, there's the age of the population, there's people with pre-existing conditions, there's people who are immunocompromised. So animal models are useful, necessary, valuable, but don't ever believe that they're a predictor of the reality of what you're going to see in humans. And sometimes, there's always the argument, well, non-human primates are better than mice. And sometimes yes, and sometimes no, because they're just a model, just like, every other model. So nobody would pretend that the preclinical models are predictors, but nobody would, I don't believe, say that you don't need them. So criticizing them is stating the obvious. Trying to use them rationally is the path forward.
Yeah, thank you. There's another question that.
Sorry.
Go ahead, please go ahead.
I would like to speak. So I agree with everything that Derek just said. I would add to that one of the challenges with especially with rodent studies is the size of the animal, right? So we often use a tenth of a human dose of a vaccine in mice, which is still a very huge dose compared to the size of the animal. And I think that probably introduces some just mechanically more damage at the injection site, which may influence the immune response. And that's why I think I would argue that it may be important to do more studies to see whether, for example, mini pigs are a better model for prediction of the vaccine outcomes for certain, at least for certain vaccines.
That's absolutely true. That's what you find out as you go forward in from your preclinical to humans. You learn these lessons. Another thing that
I think,
go ahead, Rajesh.
I think it may be worthwhile to look at some of the veterinary vaccines where you can study with and without adjuvants in large animals. I have not looked into the veterinary vaccines, but maybe there are some people who are from the veterinary vaccines field and they can share their experience that if the aluminum adjuvants are really working as adjuvants. I think animal models, as Derek said, that they are necessary to start a project. You have to show some proof of concept. You cannot jump into the humans without any basic proof of concept. So, but relying on those going for too long, I don't think that's worth it. And then the ultimate, it is the humans. And I think the veterinary studies, veterinary vaccines should provide some information on that.
I think use of various adjuvants in veterinary vaccines is also getting to be more popular today. So I think a good example that I thought that we have here is Corbevax. That during the COVID, it was kind of a bold step to take that combination quickly into the clinic, and obviously it worked very well. So that's a very good example that we have from recent development. Another thing that we run into and is that some antigens require aluminum to get an immune response in mice or rabbits. But in humans, you actually don't need the adjuvant. You don't need even aluminum. And sometimes, so when you regulatory preclinical write-up, you always run into problem. You have chosen to go in without an adjuvant, but your preclinical studies have aluminum. And to justify why I don't need aluminum in my clinical study gets to be very complicated, because you're basing it on obviously understanding of the antigen from other experiences and other clinical studies, but your preclinical models that you really need to use for your work, it's difficult to get any response without the aluminum. So you always run into all different challenges with preclinical models. Other comments, questions?
I have also a comment about these immunomodulators, TLR type of adjuvants, which seem to be working very well. So I think the only question I have is that they are very potent adjuvants, and hopefully enough studies are done. There need to be a balance between their side effects and the adjuvant effect, because they have tendency to go producing sometime antibodies to MPL. As I mentioned in my presentation briefly, that MPL produce antibodies to lipid A, and there is some literature from 90s that lipid A antibodies cross-react with human muscles. So there may be potential for some autoimmune diseases in certain individuals or others. So, and the other thing is that I think the nature has built a safety net that when we age, our immune system slows because we are more prone to autoimmune diseases. So if we try to use very potent adjuvants, then you have to balance that risk against that getting the autoimmune diseases or producing antibodies to your own muscles or other organs. That's what I caution based on my experience and knowledge about the adjuvants. I'm not saying that I think these talks have been excellent and they are moving in the right direction, but just putting a caution that this issue should be looked at carefully.
Of course it is. So I'm in a company who've been developing novel adjuvants for a couple of decades. Essentially, those products have been extensively scrutinized by the agencies during licensure and with post-licensure. And I already highlighted the risk benefit is established. So I don't think speculation about what may happen or may not happen really helps the case. The data is available, the data is published, and the data is within the agencies who have the necessary oversight and scrutiny.
I don't think it is speculation. I think I don't want to go into that debate, maybe at some other time. But there are some cases of like having autoimmune kind of reactions after even Shingrix vaccine or others. So I don't want to go into details at this time. But I'm sure that GSK and FDA looked at all that information and they approved it based on the risk benefits, and the probably benefits outweigh any perceived risk. But as I said that I support what is being done, but I just put as a caution based on I think my association with the adjuvants for almost 40 years, where even we published that for adjuvants, the balance between the reactogenicity and adjuvanticity has to be maintained, which is basically a risk-benefit analysis.
We touched upon the aluminum formulations, etc. But I think one of the things that I also wanted to raise in my own experience is the aluminum formulations. You can run into a lot of problems, especially in the fill finish process, because I think somebody did mention about the particle size, and the as you scale up processes for fill finish, your mixing reaction, etc., will impact your particle size, and then when it gets smaller, they start to get clump. and you start to have problems after you filled your vaccine, the vaccine clumping, leading to visual inspection problems. Has anyone encountered any of those? Problems with fill finish?
I might take this answer. I mean, actually, I'm replying to a similar question there on the chat box, and it's true that we have a number of customers always coming back to us at Croda Pharma to with these kind of observations and comments, and then asking, okay, what is happening? So it's clear that with some formulations, as you add, relatively complex mixtures, in particular with proteins, and that's the essence of my answer that I will finish writing. These proteins we know will adsorb to the surface and then change completely the surface properties of the particles with adsorbed proteins, and that can lead to, as you have seen through the talks, to protein denaturation on the surface, and therefore exposure of hydrophobic patches, which will then trigger aggregation of the material and eventually formation of these visible particles, which are in excess of hundreds of micrometers. So that's the mechanisms. It can be also simply or excipients also in the formulation. So I guess that the really want us to decompose these different effects to if this is observed, if this is experienced, meaning further aggregation or formation of particular aggregates upon formulation, to look at the different components and see how they affect effectively the behavior of these initial flux of allyl hydrogel or Adjuvants or other aluminum oxides. So that can be investigated through dedicated experiments, and then to try to get an answer. I mean, if your protein is effectively destabilized on the surface, where it's likely that further aggregation is going to happen. Actually, that happens for some proteins on their own. If you denature them, they will tend to aggregate and then form these eventually these visible flux. So I would recommend to investigate these, let's say protein alone, what is known about its stability, protein plus the adjuvant, what is happening, and eventually the components in the rest of the components in the formulation. I mean, does it, let's say, alter significantly the surface properties of the adjuvant? So that's for the global problem. Now, after fill finish, I think there's a nice series of articles where Chris has been involved, showing the use of NMR to actually study, sedimentation and in the vials. This is of course applicable for small volumes. It's probably difficult to do it in large containers, but at least it demonstrates that after the fill finish, once the container is effectively sealed, one has still the opportunity to look and to actually to understand what is happening after the fill finish is complete, and in particular to look at the stability of the individual vials. I think this is actually quite remarkable and should be exploited.
Yes, definitely, because there is back at Pfizer, we did do a lot of work on understanding this process, especially on the fill finish, especially on the filling, the mixing speeds, and the recirculation, how each of these factors impact to be able to ensure that you don't have this the aggregation phenomena occurring, and maintaining the particle size. So I take your feedback. It's very important for manufacturers. I do see that there are questions on the chat. I think one is.
I would like to also comment on the segregation issue. Like without naming the vaccine, I know that the initial formulation of a vaccine had those kind of aggregates, and the gel would settle very fast. So I think if you have those problems, you need to work on the formulation. Like as Lakshmi mentioned, like at how they are mixed, and maybe you may need to include some like detergent polysorbate 80, polysorbate 20, or other excipients to optimize your formulation. Because what happens that with the aluminum, if the vaccine antigen bind to multiple aluminum particles, then they tend to become bigger and they start to precipitate. And this can also happen with the whole cell pertussis vaccine. I think somebody asked about the DTP vaccine. So the formulation needs to be optimized, and which is unfortunately lacking in many of the old vaccines. They were not doing much formulation. But for newer vaccines, Lakshmi knows that how much time they were spending on the formulation development at Wyeth. And I think that's what it is needed.
We have questions from Serum Institute on, is it right in understanding that desorbed antigens may not show the same potency in vivo ELISA test as opposed to antigens used without adsorption?
I think they mean in vitro ELISA. I don't know. It says in vivo ELISA.
Oh. Vamsi, could you speak up to your question? Are you talking about an in vitro ELISA test? Following desorption of your vaccine?
Yes. I'm sorry. Sorry for the spelling mistake. It's in vitro.
In vitro.
Thank you. My question is more directed that
how do you desorb the antigen? Actually, it depends upon that. How do you desorb, and how much like desorption is? Is it complete desorptions? So there are ways to solubilize the gels, which may sometimes affect the epitopes or antigenicity, or there are some gentle ways. And again, it depends on what kind of adjuvant. As I mentioned that for aluminum hydroxide, if it is ligand exchange, it is difficult to even dissolve the adjuvant. So it will be like case by case if you know the specific what adjuvant it is, what antigens. So you will develop a process to desorb it as much antigen as possible. So if you cannot dissolve completely, then of course you are going to get a lower potency as compared to the soluble antigen.
So, Bob, do you want to comment on doing ELISAs with the on aluminum, the vaccine on aluminum for the potency assays, which may be a better way? The experience with NRRV. Bob? I think we lost Bob. But there are some publications that are available that talk about doing the potency assays for the antigens with the on adsorbed onto aluminum. So you may want to look up some of those references, which might be, if it works for your particular antigen, then it definitely is a better way to go than having to desorb even. Because yes, it is always a challenge. Solubilizing, you could lose epitopes like Rajesh mentioned, or with something like aluminum hydroxide, it's very difficult to get it off completely. And the time it takes sometimes to get it off, or the concentrations of buffers that you need are very high. So sometimes it's not practical. So it's always a challenge. But
I have also done some work on the trying to quantitate antigen while adsorbed. So it does not work with every antigen. I can say that. I think you have to look at your antigen and whether it is binding to your epitopes you are looking for. So it depends. I'm not saying that it is going to work with every antigen. So it is again case by case.
Isab, did your question get answered? Because we did talk about the particles. And Biopharma had a question.
Yes. Thank you. It's already answered.
Okay. And Vamsi, you have another question on Bob's presentation. Did we lose Bob?
Yes, I think so.
Okay. Definitely we can ask him to respond to it and get back to you.
Sure. Thank you, ma'am.
Yeah.
Like,
I'm just trying to see.
if I
Yes.
may, in the case of particle formation, has anyone experience finding particles when we formulate adjuvanted vaccines with thimerosal as a preservative? Is that something that can trigger the particle formation as well? Thank you.
I have heard about this, but yeah, I have heard from some cases that with the thimerosal sometimes the particles appear. So I don't know the mechanism, but I'm aware about this issue. Again, the solution is the formulation. Probably you have to go back to the formulation and that to include some detergents or something so that there are no particles.
I have another question from Amit Shirodev on analytical methods to determine antigen content once adsorbed on aluminum hydroxide. So you're talking like a potency assay. Yes, there are some publications that are there. If you have good antibodies that you may use and try it out. But it's all, if it's a single antigen and if you're looking at protein content or something, that's probably simpler. But if you're looking at a potency type of assay, I think you may want to try some of these assays that are there in the in literature. Any other questions? This is your chance to ask. You have all the experts here. I know there has been questions that have come up at PATH around, especially this is for Lauren, for you, from CRODA, about the re-sterilization of aluminum or even the containers that are available. And I was hoping that somebody would bring up some of those questions, but I guess we lost some of our key people
Audience
because of the time zone.
Well, as you actually asked, so of course we know it's a major issue, and as I've presented, we know that every time there's a new cycle of heat or heat cycle through autoclavation, for example, that has been demonstrated, and that the material properties will change, and moving from let's say a given amorphous phase to a more crystalline phase with the effects that I've described, which means a reduction of the surface area, therefore the number of binding sites, and so on and so forth. So how to avoid this? What do we do at CRODA with this? So I think that the solutions, which are technical solutions, exist, and that's simply to effectively put on the containers sterile connectors, which can then be coupled directly at the client, at the customer, the vaccine manufacturer, to effectively do a sterile connection with their chain, their own production chain. This necessitates. So we are working with these issues. There's a team working with this. There's still debate of which type of connector and so on. And then the biggest question is how mature is the industry to effectively accept standards, because you understand well that we do not have, if we go this way, which we hope we do, at least with selected customers at the start, we cannot provide different standards. This is going to be already quite expensive to implement, which will come with an additional cost to the product when you start to couple or to use these sterile connectors to the containers, and then if on top of that we have to case by case have specific connectors, that's not going to work. So it means that it's a more global discussion with the different customers, vaccine manufacturers, to know if what kind of standard could be effectively implemented to ensure that this is not an issue anymore. Because then the goal would be to, okay, we guarantee sterility. There is effectively a steroid connector. You just plug in, pull the membrane, and then you have transfer directly into your first step without having to re-autoclave. And we know that re-autoclaving does effectively affect the properties of the material. So that's effectively a big question for the whole business in general, not only for us. We are actively working with this, and we hope that we'll be able to provide some solutions in the near future.
I think it is not recommended from the regulatory perspective also. Actually, you should not be re-autoclaving unless it is part of your manufacturing process. And secondly, I don't recommend it. I think the thing is that I don't know why there are concerns. There are other ways to address these concerns. There should be shipping validations. Like when they ship these containers, they should be validated that they have container closure integrity and the sterility is maintained. And then there is a aseptic process validation anyway. When you formulate with the vaccine, that process cannot be like autoclaved. And another point is that autoclaving aluminum adjuvants is not that straightforward. Somebody who has experience in autoclaving, Lakshmi, you know that the heat does not penetrate and reaches the center. So I don't know why, from where this question is coming. It has a lot of regulatory implications. You are making a new product. You may be asked to go back to do clinical trials that with the extra cycle of autoclaving, your vaccine is still efficacious. So I have concern about these question, and I think there are other ways to address it. If there are shipping validations and the aseptic process validations. So I don't know. I know that you asked this question during our pre-meetings also.
This actually needs to be addressed even early on in the process, prior to even phase 3, because that's when you're actually scaling up your process and getting ready to and thinking about all the elements that are required. And one of the ways we've always looked at is, after you've sterilized it multiple times, looking at the particle size, looking at its binding characteristics, addressing all these factors that are important for your vaccine formulation before you dive into the actual formulation for your scale-up vaccine. Because many times I think the issue that folks face is the container of aluminum that you get. If you get a large container, then you're not going to utilize all of it, and it gets to waste if you have only used it for one formulation, or if you have taken out aluminum from that particular container, you need to go back, do sterility testing. There are multiple things that needs to be addressed. So from a manufacturing perspective, there are a lot of questions that need to be thought about. I see a couple of questions. One is on the critical, how critical is stirring during al-hydrogel formulations for adsorption during multiple interactions?
It is not clear what is multiple interaction, like adding multiple antigens, or the question is not clear. Maybe the person can speak up.
Gaurav, can you speak up to what you're looking for in terms of multiple interactions? Are you talking about multiple, doing it multiple times, or?
So there are multiple ways the antigen and gel can interact by hydrophobic interaction or by ionic interaction, like that.
No, but that does not matter. I think everybody showed that in the end, the most important is the ionic. And even that, when you make a vaccine and you add other antigens, they are going to rearrange. So I don't think that is going to matter. Once you have a manufacturing process, you set up some range of the adsorption and show consistency in manufacture.
Yes, your mixing or your stirring is important, but you have to develop your process to ensure that it is it doesn't disturb the characteristics of your aluminum, your adjuvant, multiple things that you need to look into. There's another question that I see Laurent has given an explanation. And one more question is about what is your view on formulating alum adjuvanted vaccine in a microarray patch?
I don't know. My knowledge about microarray patch is that you are talking about that skin patches for vaccines,
Yes,
or
yes, those are the skin patches.
so I don't know. I think if the particle becomes bigger, I don't know how it will pass to the skin cells or others. So my understanding.
My understanding is that. Oh, sorry. My understanding is that aluminum adjuvants are not recommended for intradermal vaccines because they are thought to be too reactogenic. It's not more reactogenic than in the muscle, but it's more visible that the reactions to the vaccine. And so I think the general recommendation is to not use aluminum adjuvants for intradermal vaccines.
Yeah,
that's true. But also, like if it is a patch, the patch is that the mechanism it works that the vaccine absorbs into the skin cells or other like immune cells. So I don't know if the something absorbed on through the aluminum particles will be absorbed as efficiently. I think usually with such vaccines, there are some other adjuvants like cholera toxin or others which are soluble are used. I have never heard the use of aluminum adjuvants for skin patches.
Yeah, and on top of Harm and Rajesh comments, it's clear that you have already a practical problem, given the fact that these materials are existing in at least standard gel or similar, aluminum hydroxide exist in flocks of large aggregates, which are, up to 40, 50, 100 microns sometimes. And this material does sediment, and it's already effectively a problem in normal dose, in particular for two-component vaccines where you have the aluminum adjuvant, which is kept apart, and then you have to resuspend it. So you can imagine that in these microneedles, you might have effectively very easy clogging of the
yes, exactly.
the needles, and that's it. So then I would recommend to consider some of Chris Fox's nano alums for doing this. But then we are back to Harm's comment about the reactogenicity at the injection site, if it's subdermal or intradermal.
And you can play with the aluminum adjuvant. Actually, you can add phosphate or reduce its ionic charge or other things. I think there are a lot of ways. I think I liked the aspect after studies done by, I think, Professor Hem and his colleagues, that you can change the point of zero charge. So it becomes very versatile and will help you to optimize your formulation. So you have to do it scientifically. And I think it's a very versatile. Now, if you look from that angle, and you can get what you want on a wide range of pH.
I think we are over time now, and thank you so much, all of you, for having taken the time to make these presentations and present here, because this is going to be very valuable. We are going to put this on a website, or we will figure out how to make it accessible to folks who are looking for the presentation or who have not been able to participate because of the time zone, etc. And there will be folks who will be asking for it. So really sincerely from Path, thank you so much. And this has been really valuable. And I hope we can do something like this again. This has been really exciting personally for me. So any last minute? Any last minute from anybody before we close? Okay. Laura, anything last minute?
Nothing for me. Thanks everyone for joining.
Okay, thanks everybody for joining and all the speakers for the presentations. Yeah. Thank you again.